It's my absolutely great pleasure to introduce our plenary speaker, Susan Hagness. Susan is a professor of, for those of you who don't know, Susan is the Philip Reed Professor of Electrical and Computer Engineering at the University of Wisconsin-Madison. And she has been a longtime contributor to URSI. She's a fellow of a number of societies. I'll point out IEEE and AAAS. Susan's research has been in computational electromagnetics, focused on bioelectromagnetic problems, and I'm a big fan of her work in breast cancer detection. She has an amazing publication record, over 100 journal papers, several book chapters, and a very well-adopted book in FDTD. Over 40,000 citations, an amazing H-index of almost 60. So she's really accomplished in research, not just in research. She's also a fantastic teacher and mentor and has been recognized by a number of awards. We only have an hour, so I'm not going to list all her awards, but I'll point out the NSF Case Award and also the URSI Issa Koga Gold Medal, which is a very prestigious, only given every 3 years. Susan is also a fantastic citizen. She's done a lot for URSI and other societies, but I want to also point out that she's currently the department chair at the University of Madison in Wisconsin, and she's very committed to diversity. Her department has an amazing number of 25% faculty who are women. She's always been promoting women in science and in engineering, including actually raising a daughter who will be studying physics. And so with this, I'd like to welcome Susan Hagness, and we're very excited to hear your talk, Susan. Always learning a lot from you.
Zoya, thank you so much for that really affirming introduction. I appreciate it. I'm delighted to be here. It has been a couple of years since I've been out to Boulder for this January meeting, and just really thrilled to be here with you all. So I want to say a special thank you to the USNC-URSI Women in Radio Science chapter, in particular the officers from the first 3 years. When I reflect over the last 70 years, what progress we've made. We still have a lot of progress to go, but this photo on the left shows you the 10th General Assembly of URSI back in 1952, and the first woman radio astronomer, Ruby Payne Scott, in a large group of men. And now we have, a room full of a gender diverse community. And so again, we've seen just tremendous progress, and I'm grateful to the leaders of the Women in Science, Radio Science chapter for the invitation to be here with you today. I also want to take a moment to remember Pat. As I understand, she was originally slated to be standing here with you today, and so I'm humbled to be filling those very big shoes. She worked tirelessly to increase the participation of women in radio science, and we can grieve her loss, but also celebrate everything that she did in her career professionally, both technically and for encouraging diversity. So the overview for my talk today is this concept of radio science meeting both smart medicine and smart agriculture. So we're going to take a tour of some of the fundamental microwave interactions with tissue, both human tissue, animal tissue, and plant tissue, and look at how we can combine and exploit those fundamental characteristics with some sensing technology, some machine learning, to achieve some technological advances in both medical applications and agricultural applications. And here by smart, I mean something along the order of adaptive or autonomous, or just generally speaking, technology enhanced. And so I'm going to focus on 2 case studies. One involves machine learning enhanced microwave estimation of cranberry crop yield. That'll be the second case study that I get to, but we're going to kick things off with a deeper dive into some image-guided microwave ablation of tumors. But before I do that, I want to set the stage with an overview of electromagnetic interactions with tissue. And what are the fundamental mechanisms that are at play here that allow us to develop some really exciting technologies? So along the electromagnetic spectrum, if we go high in frequency, the electromagnetic waves are interacting with the materials at the atomic level. And if we move lower in frequencies down into the microwave and RF range, the electromagnetic energy is interacting at the molecular stage. And whereas at atomic level interactions, we're exploiting some of the fundamental characteristics of tissue that involve electronic bonds. In the microwave frequency range, we're exploiting some of the fundamental characteristics that have to do with permanent dipole relaxation and ionic bond, molecular-based ionic bond resonances. And so, if you think about tissue being primarily comprised of water, we've got an inherent mechanism there for correlating the macroscopic dielectric properties at microwave frequencies with physiological content of tissue. And this has to do with differences in free and bound water content, protein content, all of the types of permanent dipoles that, or induced dipoles that can exist in tissue with macromolecules, amino acids and the like. And then a variety of physiological changes that can take place with disease, temperature changes, or through treatment, temperature changes, inflammatory processes and the like. These can all influence the macroscopic dielectric properties. And so I want to highlight some of those dielectric properties contrast that we can exploit. And again, we're going to look at animal, human, and plant tissue. So I mean biological in the broadest sense. So this slide shows you some highlights from a canonical study published by Gabriel and Lau and Gabriel. And it illustrates, I pulled some of the representative tissue types from that trio of papers that was published in Physics and Medicine and Biology. And you can see over a very large frequency range, this incredible soft tissue contrast that exists in both the dielectric constant and the effective conductivity. These dielectric properties of biological tissue are measured oftentimes using localized measurements with coaxial probes. And so in this case, an open-ended coaxial probe is placed in contact with the tissue. The fringing fields at the end of that open-ended coax interact with the tissue. The tissue can be treated as the material under test, assumed to be homogeneous within the sensing volume of the probe. And then the average dielectric properties within that sensing volume are extracted by measuring the reflection coefficient at the aperture and then converting that to the unknown dielectric properties. And these properties can be measured with laboratory probes, like a precision probe that we developed many years ago with the University of Calgary. There are clinical applications that use dielectric probes for characterizing tissue for clinical purposes. And over the years, we've had the opportunity to characterize some of the properties and study how the impact of certain physiological conditions can impact the dielectric properties. So as one example, we looked at liver tissue and what's the impact of temperature. This is of interest for microwave thermal applications in medicine, such as microwave ablation. And so again, you can see that over a wide frequency range as temperature increases, because of what's happening with those dipoles in tissue and how they can respond under different temperatures to an applied electromagnetic field, we've got varying dielectric properties. In particular, if we look at the UHF band, below 50 degrees C, we've got a modest dependence on temperature, but above 50 degrees C, there's a significant dependence. And we can exploit this, as I'll show you a little bit later in this talk. Breast tissue, the dielectric properties are dependent on composition of breast tissue. This is a particular type of tissue in the body that's very heterogeneous. And so depending on whether there's a lot of fibroglandular tissue or mostly fatty tissue, the dielectric properties can really be all over the map. So normal breast tissue properties, what we found, depend strongly on the amount of fat that's in the tissue. We've also cataloged the dielectric properties of malignant breast tissue relative to a healthy breast tissue and observed about a 10% contrast. So composition matters as well. The impact of ablation. What is microwave ablation? When tissue is heated to levels where coagulation and necrosis occurs, what impact does that have on the dielectric properties? Well, we did a study on mastectomy specimens where we performed an ablation and tracked the impact of ablation on dielectric properties, so before and after ablation, and saw that there's a significant contrast between ablated and non-ablated tissue properties. Relative permittivity drops by about 40% in the study that we did, where we were ablating primarily fibroglandular tissue. So the gray curves here are the healthy reference. The damaged tissue after ablation is shown in red. And then as an example of a botanical target, we've looked at the dielectric properties of the fleshy tissue, the cranberries, fruits in cranberry crops. And so here, we're looking at the impact of composition, again, in this case, across the vine, the plant, the full plant. So as a reference, water is shown here on these graphs of relative permittivity and effective conductivity over frequency. And the cranberries themselves are high water content. And that's why when you think about cranberry crops, you think about what it looks like at harvest time when the cranberries rise to the top of the flooded bog. They're full of a lot of water, and so they have high dielectric properties. The leaves, if they're healthy, they have about a 3-to-1 contrast with the berries themselves, and dried out leaves, which end up at the bottom of the field above the soil, those obviously have essentially no water content, so very low dielectric properties. So significant contrast between the berries and the fresh leaves. And we're going to see how we can exploit these contrasts for some interesting technological applications. And first up is the topic of microwave theranostics. And what I mean there is the combination of therapy and some diagnostic interventions to track the progress of therapy. So we're looking at ablating tumors with heat. And this is one of the interesting multiphysics effects when EM, electromagnetic energy, is absorbed in tissue. So from the Penny's bioheat equation, we can see that if we have electromagnetic energy absorbed in tissue, that serves as a source term in the heat equation. And we can achieve significant temperature rises when we deposit electromagnetic energy in tissue, as illustrated with this image of an ablated liver with an interstitial antenna that's been inserted into the tissue. And you can see that in the region right around the antenna, the tissue has gone through the highest level of heating. And so it looks like cooked meat. And as we move away from the antenna, there's been less deposition of microwave energy. And it looks like, if you've ever looked at uncooked liver, that's what it looks like. So what is the current state of microwave ablation technology? It's primarily an intervention that uses interstitial microwave antennas that are inserted into the tumor in one of 3 ways: percutaneously, in open surgery, or laparoscopically. And microwave ablation technology is available commercially. There was a startup out of Wisconsin that turned into a new wave that was then bought out by Johnson & Johnson. And our surgeons at the University of Wisconsin Hospital are using microwave ablation technology on a routine basis. Here's an example from Ethicon, the arm of Johnson & Johnson that produces microwave ablation systems. An image of a probe placement scan. So there's an example of an antenna that's being inserted into a lesion in the lung. Now, our vision has been, well, can we move further and offer a catheter-based means of ablating tumors anywhere in the body? And so to do that, we need to add some additional tools into the ablation toolbox. The vast majority of interstitial antennas in the past have been designed to operate at around or below 2.5 gigahertz. And so those large, lower frequencies impact the length. Question is, can we go to shorter antennas with higher frequency operation? We've looked at that. Vast majority of interstitial antennas are based on coaxial cables, and those require balance traditionally. That allows us to achieve localized heating, but increases the diameter of the ablation antenna. And so the question that we've been asking over the last several years is, can we achieve narrower diameters with innovative antenna designs? And then finally, these coax antennas are rigid typically. And so they require line of sight or direct linear access to the tumor. And we've been exploring in collaboration with an industrial partner whether we can offer flexibility and get to that fourth realm of catheter-based, routing to a tumor with, again, some innovative antenna designs. And so today I'm not going to focus on some of the work that we've done on higher frequency microwave ablation or work that we've done on kind of getting towards narrower diameter antennas with reduced diameter balance. I'm going to go straight to a couple of examples of balun-free antennas and then how we've been able to achieve some initial progress with flexible antennas. So the first type of antenna that we have proposed and demonstrated is a base-fed, a balun-free base-fed helical monopole. So if we were operating this antenna at the fundamental resonance without a balun, we'd get a specific absorption rate like what you see on the left. And in that case, the power absorption is occurring all the way down the coaxial feed line. And that results in an ablation pattern that's not at all what we want. It's too spread out. But if we move to the second resonant frequency of the antenna, so that this is a half wavelength helical monopole, then we are operating at a frequency where the current is a minimum at the feed point. And so we get a high input impedance at that operating frequency that effectively chokes the currents that would otherwise want to flow on the outer surface of the outer conductor of the coax feed line. And so with that operating frequency, we can achieve without a balun a very compact specific absorption rate pattern or SAR pattern of power deposition, as you see with the graph on the right, the lower right. But because we're operating at a frequency where the input impedance is very high, we have to implement an impedance matching network. And we've done that with quarter wave transformers or coaxially implemented pi networks with reactive elements. That works fine with rigid or semi-rigid antennas, and we can achieve compact ablation zones there. But it precludes moving to a physically flexible design. And so we've also looked at balun-free antennas that are made up of helical dipoles, where we take the inner conductor of the coax feed line and extend that to form one arm of the antenna. And then we take a portion of the outer conductor and extend that and wrap it around and create a helix to form the second arm of the dipole. The current on that outer spring or helix, I call it a spring because we've implemented it with a spring. The current it reverses its direction a quarter wavelength away from the feed point, and it ends up aligning with the current that's on the inner extended conductor. And this creates a relatively low input impedance and chokes the current that would otherwise flow on that outer surface of the outer conductor. So without a balun, without an impedance matching network, we get a compact SAR pattern, a compact ablation zone. And we've tested the maneuverability of this because this particular design is amenable to being implemented in a flexible coax. We can go to a very narrow diameter cable. This example is 1.64 millimeters in diameter. And as you see on the right, we've demonstrated running it through a test fixture that bends at 180 degrees. It doesn't have an impact on the performance of the antenna. And we've ablated a perfused liver with this flexible antenna, as you see here in the photographs on the right. Now, being able to get to a tumor through a robotically assisted catheter routing mechanism is exciting, but we also need real-time guidance to track the evolution of the ablation zone. And the current strategy is to take microwave ablation and pair it with an image guidance tool like MRI or CT or ultrasound. We've been looking at ways in which we could integrate the image guidance directly with the microwave ablation technology itself. The rationale is to exploit the dielectric properties changes or other types of changes in the tissue properties during ablation, and to also exploit the presence of the interstitial antenna itself as a source. So I'm going to show you two examples, one in which we use the antenna itself as it's transmitting energy to ablate. We can also use those signals to then pick up some information-rich signals at the surface of the tissue
and determine the boundaries of the ablation zone in real time. Alternatively, we can exploit a multi-physics effect that I'm going to explain in more detail in just a little bit here. We can exploit a multi-physics effect that triggers the generation of acoustic waves and then use ultrasound transducers to pick up, again, information-rich signals at the surface that will tell us some information about how the ablation zone is evolving during the treatment. So first up, I want to highlight some work that we've done on a time difference of arrival-based microwave imaging algorithm that can be implemented in real time to monitor the progress of ablation. So the data acquisition configuration is shown with this cartoon where at the beginning of ablation, there's no ablation zone. So we start with the baseline properties environment. At point A, the ablation antenna has been inserted, and point B is a future boundary as the ablation zone reaches that point. Point C is an antenna outside of the body that's going to pick up a signal that's being transmitted from the ablation antenna itself. Now, at a certain point in time later in the ablation procedure, an ablation zone has evolved such that its boundary along the path from A to C lands right at point B. So we record the signal at C at that moment in time and determine the time difference of arrival relative to the baseline through by maximizing the cross correlation. So if we know that time difference of arrival, then we've derived a very simple relationship between that time difference of arrival, which can be measured, and then some known properties, and determine where what the distance is between A and B. And if we do that for all of our receiving antennas surrounding the region of interest, then we can construct an ablation zone boundary estimation. And we've illustrated that in simulation with some 3D heterogeneously dense breast phantoms. You're seeing 3 orthogonal cross sections through the breast, and the dots or the little vertical segments are showing you the positioning of some external antennas. The open circle in the middle, right here, is the location of the ablation antenna that's creating an ablation zone of 10 millimeters by 7.5 millimeters by 10 millimeters within this complex tissue, otherwise healthy tissue environment. Ablation, as you saw from some of our earlier highlights, reduces the dielectric properties of the tissue in that ablation zone. And so this color map is showing you the dielectric properties of breast tissue, both the high properties of healthy healthy fibroglandular tissue, and the lower dielectric properties of the ablation zone in what is presumably the malignant region. And so the red dots are showing you where we've reconstructed points on the surface of the ablation zone with this technique, very simple technique that I just described. Processing time is negligible. And even at 0 dB of SNR, we've got a root mean squared error of just 1.5 millimeters in locating the boundary of the ablation zone. So that's one interesting way in which we could track in real time with some technology enhancement the evolution of ablation. The other example that we've explored is, it stems from this question of what else happens when microwave energy is absorbed in tissue. So if we introduce a pulse, a microsecond long pulse in tissue, and look at what happens with just that single pulse being absorbed, there's a micro temperature rise. If we hit the tissue with enough of these pulses, we're going to achieve an ablation zone like what you see in the photo on the right. But just one pulse will induce a slight temperature rise. That causes an expansion of the tissue volume through the thermoelastic effect. And that thermoelastic expansion launches an acoustic wave. So this is a really rich electromagnetic, heat, acoustic, multiphysics problem that we've been exploring to achieve real-time microwave ablation monitoring, where we've got a pulse microwave source instead of the conventional CW source in microwave thermal therapy, an interstitial ablation antenna that's delivering through a pulse train enough energy to ablate the tissue that at any moment in time is launching these little bursts of acoustic energy. And we can pick those up with an ultrasound transducer to try to learn something about that ablation zone. And so we have explored this concept both in simulation and experiment. The experimental setup involves a carrier frequency of 10 gigahertz, a microsecond long pulse width, peak power is 25 kilowatts, and a floating sleeve dipole antenna. You can see the configuration for this experiment in water. We used water because that's a nice homogeneous medium for validating a simulation tool, which is a multi-physics simulation tool. So you can see in the upper right, the coupling of an electromagnetic solver, a heat transfer solver, and an acoustics module. We've done this in ComSol, where information is being passed through these multiple physical solutions to track the launching and propagation of acoustic waves. And we've looked at the time of arrival of those acoustic pulses as a function of temperature of the medium in which the pulses are being launched. And so we've gotten very good agreement between simulation and experiment up to the temperatures that we were able to conduct our experiment at. We had an ultrasound transducer that wasn't rated for higher temperatures, so we didn't want to destroy it. But it gave us confidence in the simulation tool, and then it allowed us to use the simulation tool to go to some higher temperatures. All right, so with that, we have looked at one case study where we take what we can exploit about microwave interactions with tissue, combine it with some sensing technology, and hopefully move forward with some new tools in thermal therapies for tumors. Now, this is one example, one case study of what I think is just an incredibly rich and exciting field. There are talks here, and as I look around the audience, I see other researchers who have been working in this field, and I encourage the community to look in awe at some of the amazing research that's being done in biomedical applications of radioscience. Next up, I want to switch to an agricultural case study where we again look at what we can exploit in terms of microwave interactions with plant tissue, combine it with some sensing technologies, and in this case, machine learning. We're going to throw that into the mix and look at some agricultural applications. So this segment of my talk is machine learning enhanced microwave sensing of cranberry crop yield. So I'm going to take you to a cranberry field in Wisconsin. You may not know that Wisconsin is the leading producer of cranberries in the country. Everybody thinks Massachusetts, but it's actually Wisconsin. In central Wisconsin, we've got all the right environmental ingredients for cranberry crops, 21,000 acres of cranberry crops in Wisconsin. And again, the photo on the right is what I was kind of alluding to earlier with my comments. You think cranberry crops and you think beautiful lakes of cranberries floating on the water, but that only happens at harvest time. The cranberry crop for the growing season looks like what you see on the left, where you have cranberries that are growing on vines, really thick vines. And throughout the growing season, farmers want to have predictions of what their crop yield is going to be. Agricultural scientists want to have predictions of what the crop yield is going to be in their research plots as they look at different genetic variations of cranberry crops. And the current technique, like the gold standard technique in play with your typical farmer, is a laborious hand picking of all the cranberries in a 1 square foot region of the crop. And then they try to extrapolate out from that sample. Or maybe they'll do a couple hand picking rounds. But either way, it's a technique that at best gives about a 15% accuracy on the yield. So where can radioscience play a role here? Many years ago, an agricultural scientist in Wisconsin, part of Ocean Spray, came to us at Wisconsin and said, surely there's a better way here. So this community is well aware of the better way. Microwave remote sensing techniques. We've got a rich variety of options here. So in terms of sensing platforms, we can leverage ground-based or space-based or air-based platforms. passive radiometry or active radar type techniques or scatterometers. And then in terms of the signal processing that can be applied to the microwave measurements from that field, well, there are scattering models that can be used to do a full inversion to get the parameters of interest, or we can use machine learning algorithms that are statistically based, classification algorithms, regression algorithms, where ultimately we're doing some sort of statistical inference to get the estimated parameters of interest. And so in our case, we want ground-based, at least initially, ground-based or close to ground-based, because we have to validate this technique. And so we want localized measurements so that we can get the ground truth and validate. We're looking at an active technique where we're transmitting low power microwave signals into the crop. And as I've already hinted at, we're using a machine learning algorithm that's classification based. Our estimated parameter is berry yield. How many berries are in a region of interest? I need to pay homage just momentarily to, again, a huge amount of work that's been done in this community. So these are just a couple of examples that just scratch the surface of ground-based scatterometry and statistical algorithms or machine learning algorithms that have been applied. So a variety of crop types, but we had, up until the work that we had done, there hadn't been a lot of work done on cranberries relative to some of these other crop types. But you can see that there's been a variety of frequencies, a variety of polarization strategies. So what we've done is, in our proposed microwave sensing system, is deployed a system hardware that comprises a waveguide, phase stable cable, portable planar VNA, portable battery laptop. We collect microwave backscatter. We also check the soil moisture content and the canopy height for reference. And the data processing is a statistical algorithm where the signals from a particular region are fed in and out comes yield. Now, in our studies, we have also been picking cranberries so that after their measurements, we have the ground truth, so that we know what the actual, for that particular field of view, what the ground truth is in terms of berry count. And we've run some simulations to derive some design guidelines. And the simulations that we've done have been idealized plane wave illumination, a region where we have a certain berry volume fraction, so 5%, 15%, they're illustrated here. These berry regions where the berries have been randomly distributed throughout a volume, it's in a background of an air-leaf mixture, and that is all above a model of wet soil. So what these initial simulations helped us do is hone in on the frequency range of interest. So in these simulations, we have fixed leaf volume fraction, fixed the canopy thickness, fixed the soil moisture, and looked at the impact of berry volume fraction. We ran simulations with 50 berry distributions to get the randomness check there, and saw that the berry volume fractions are distinguishable at frequencies below 2 gigahertz. Above 2 gigahertz, that gets us above, sort of into the range where there's me scattering resonances, and in that case, the actual distribution of berries has too much of an impact. We want to be looking sort of like at a macroscopic average and not paying attention to where this berries specifically are in the illumination region. So below 2 gigahertz is where we are operating. And you can see that the backscatter power, for the most part, is well above minus 20 dB, suggesting that we can achieve this technology with low power microwave radar. Prototype sensor is a waveguide that operates between 600 megahertz and 1300 megahertz. It's mounted above the canopy with a PVC structure, as you see on the right. And it has a compact sensing volume, which enables the ground truthing. So, we need to know that we're not illuminating a huge portion of the field, because then we wouldn't know what the actual cranberry yield is in that region. So we've got a prototype sensor that has a sensing volume that's approximately 1 square foot, which aligns with then that traditional practice of picking all the berries within a 1 square foot region. So one of my graduate students had the very unique experience of spending a lot of time in some of our cranberry fields in central Wisconsin, collecting microwave measurements over 3 years of his Ph.D. And picking a lot of berries. So we had a lot of berries in our refrigerator. Refrigerators in our labs have over the years been filled with bovine liver, cranberries, not all the same refrigerator and not the refrigerator that the lunches go into. But we've also picked up, again, soil moisture measurements, canopy height measurements, and photos as well. So it's been a large field data collection campaign in a variety of both commercial plots with ocean spray and also some research plots. We've been to 13 beds, over 200 sites within those beds, and collected over 2,500 signals, microwave backscattered signals. We train and test the machine learning algorithm, just check on time, with, as illustrated here. So we take all of the signals that we have for a particular study and divide them randomly into two sets. And we train on half the signals, and then we test on the other half. So the training is done with half the signals. We're in that training set, we've got L distinct yields. The training is done with a principal component analysis and a linear discriminant. We then use those in the testing where we take a single reflection coefficient or some averaged reflection coefficients from a particular site with a known yield, M. We run that through the machine learning algorithm and get the estimated yield. Okay, so I'm going to highlight some results that we published in last year in the Transactions on Geoscience and Remote Sensing that looked at 2 combined test beds. In those test beds, we had total berry yields within the 1 square foot regions, ranging all over the map from very low berry content all the way up to over 600 grams of berries within the illumination region. And the graph on the right shows you the percentage of test cases that had an estimation error within a certain region. And so you can see that 81% of our test cases had an estimation error that was less than 15 berries. And we did 40 permutations of training and testing with 120 signals each to make sure that we had statistical robustness in the study. So we looked at what the average was of all estimation errors for each test bed. And that turned out to be, if we looked at multiple sites, that turned out to be less than 3 grams. 3 grams is 1.3% of the average yield in one of the test beds, and it was less than 1% of the average yield in the other test bed. This is a much less, in terms of looking at a whole crop, this is much less uncertainty than the best case scenario of a 15% uncertainty reported for hand picking and extrapolating. So this tour that I've taken us on today has looked at 2 applications of radio science concepts. We've looked at a medical application involving image-guided microwave ablation of tumors. We've looked at a sensing application, one of many sensing applications in agriculture. I want to acknowledge my current group of PhD students, all of the many UW-CEM, that's cross-disciplinary electromagnetics alumni, including all of the graduates whose work I've highlighted here today. I think one of the exciting things about the type of work that we're doing in my group is these are really interesting cross-disciplinary applications. The opportunity to partner with breast surgeons, with interventional radiologists, with agricultural scientists at the University of Wisconsin has been really professionally life-giving for us involved in these research projects. I also want to acknowledge our collaborators, both at the University of Wisconsin and across the globe. Many more collaborators, but again, I'm focusing on the names of collaborators that were involved in some of the work that I've highlighted here today. So with that, I want to express my deep gratitude for you all for the opportunity to share some of the highlights from our work at Wisconsin, and I'd be happy to take some questions.
Thank you, Susan. Can you hear me? Thank you, Susan. This was a very interesting and very inspiring talk. I have tons of questions, but I can ask you later. So let's ask our audience. I believe the gentleman in the blue top. was first. Yeah, if you could use the microphones, this is a pretty big room.
I think it's on, right? Okay, great talk, thank you. My question is about the first application of radioscience in the for the ablation. The ablation zone that you showed, according to what it looked like on your slides, kind of closely resembles that antenna pattern. But I was wondering if in the heterogeneous tissue environment in the multi-physics problem that the different tissues also have different thermal conductivity. So that might give rise to a non-trivial looking ablation zone. Has that been considered as a negligible effect?
Yeah, that's a great question. Thank you for that. So one of the reminders that we've had to keep revisiting in this work is that we could have exquisitely spatially complex power deposition patterns. And we see that in heterogeneous tissue because the higher conductivity tissue is going to absorb more of the power. So we can have SAR patterns that are very unusual in shape. But once that energy is absorbed and heat diffusion takes over, heat diffusion, I think of it as a low pass spatial filter. So even though with power absorption, we can get really elaborate spatial patterns, with the actual heating pattern, the ablation zones tend to be spherical or elongated, like ellipsoids. Now, there's an exception to that, and that has to do with heat sinks. So if there are like large, in the liver, the portal veins, if you've got a vein that's running through, then the ablation zone kind of, has to wrap around that, and that can end up introducing some perturbations in the heating. But otherwise, I would say no, we don't generally, even in heterogeneous tissue, unless you're talking about like auxiliary hotspots, within the primary target zone, we don't see funky ablation geometries.
Very good. Any other questions? Can I follow up on this question?
Yeah.
So how much power do you use for the ablation?
Yeah, well, it very much depends on the particular target. So in the ablations that we performed on mastectomy specimens, where we were inserting the antenna into fibroglandular tissue, to ablate that tissue, we were well above 50 watts.
And what about the liver?
And in the liver, we were able to get ablation zones, reasonably sized ablation zones with anywhere from 5 watts up to about 20 watts of delivered power.
And so when you heat this up, you showed that the electrical properties change.
Yeah.
How do you handle that from the power amplifier side? Do you do you have a circulator, or do you do some clever impedance matching?
Yeah, great question. So,
Well, that's my side.
Yeah, that's a great question. So yes, interestingly, the dielectric properties of the ablated tissue change over time, but we have not seen substantial changes in the reflected power. So we monitor the reflected power, for example, and the
That's surprising.
Yeah.
And what about impedance? Do you monitor impedance or just power?
We've been monitoring reflected power. Oh no, we've monitored S11.
Complex.
Yeah, complex. Yeah. So we're able to continue to deliver power to the tissue in part because the changes are not happening like throughout all of space immediately. So if you think about like what's influencing the input impedance, so initially you cook the tissue right at the active point of the antenna, but there's still some higher water content
because
tissue
the tissue is part of your antenna.
Tissue is part, and it's a large region of tissue that's part of the antenna.
Thank you. Al has a question. I don't want to hog.
Thank you. And again, very, very fascinating work and very useful work too. Do you monitor at all the temperature of the ablation zone? And how would you do that perhaps through radiometry?
Yeah, great question. Thanks, Al. We've not monitored with radiometry. We have monitored with optical temperature probes that are attached to the ablation antenna itself. So we have inserted, along with the ablation antenna, a temperature probe. And in some cases, in some of our experiments, we've inserted multiple temperature probes and tracked the temperature during the ablation. And, we can see rapid temperature rises over a very short amount of time. And then, the kind of temperature as we hit more of an equilibrium state, then the temperature kind of levels off.
What sort of temperature rise do you need?
Yeah, another great question. So I didn't clarify up front, in the context of ablation as a thermal therapy, we're looking to get up to temperatures around 50 or 60 degrees Celsius or higher, so that we actually coagulate the tissue. There's a whole nother realm of applications, hyperthermia, broadly speaking, where the goal is to get the temperature up to around 43 degrees. Of course, it all depends on thermal doses, not just temperature, but also time. But, roughly speaking, 43 degrees. So, an elevation above body temperature of, maybe something like 6 degrees Celsius. So well above a fever, but, not to the level where you're literally inducing changes on the spot. But with ablation, we're trying to get 50, 60 or higher. And we observe temperatures almost up to, 90 degrees. Yeah.
Oh, sorry. I saw the young lady first. How are we doing on time, Mike?
Hi, this is a nice talk. I wonder, why do you choose S11 reflection on the cranberry case, not S21? That would be transmission method. Why do we choose reflection?
Yeah, great question. Thank you. If we were to do an S21 measurement, we would need a transmitter and receiver that are, by definition, S11 means our transmitter and receiver is co-located, right? If we go to S21, then we're illuminating from a different angle than what we're receiving at. And I think that could be, a perfectly fine way of approaching this problem. But in our research to date, we wanted to make sure that we could validate our results. And in order to validate, we wanted to be looking down at a well-defined region where we knew what the cranberry yield was for that plot. So down and up, S11. Yep.
That we have time for one more question, and I think Yaya was next.
Well, Susan, thank you very much. That was a great talk, very inspiring. Also, at UCLA, we have a lot of interest, as you mentioned. Hospitals are routinely trying to use this ablation for helping patients and all that. One of the issues we were confronted, which we are helping them a little bit, is the notion that if one use this ablation probe, usually they are fixed design. So it might work very well in liver, but not necessarily in the lung. So we were looking at the idea, potentially, maybe you are also as well, using an adaptive concept for tuning these ablation probes into the tissue as the experiments or testing are occurred. Are you also looking in this adaptive ablation probes in order to improve better performance at different locations in the body?
Yes, we are. Yes, it's a really important point, and we've noted this even. You mentioned liver versus lung. We've noted this even in the context of a particular target like breast tumors, where depending on what the environment is around that tumor, if it's a very fatty breast tissue environment, that's a very different ablation antenna design problem than if this is a very dense breast tissue environment. So looking at options with multiple different types of frequencies, geometrical features that can provide that adaptation without going into too much.
Thank you.
Okay, thank you very much. We're out of time right now, and we have a break coming up, and I'm sure Susan will be around. And if you have other questions, you can come and ask.
Absolutely.
I know I will. Yes.
Okay.
Thank
Sounds
you
good.
again. Let's thank our.
Thank you.